Hi students, I hope you have understood whatever lessons I taught so far for you. And we have some more information related to the immune system. Now let us talk about the lymphoid organs in this session. Now what are lymphoid organs? If you take the immune system consists of structurally and functionally different organs which are dispersed throughout the body. And those organs which are involved in origin, proliferation and maturation of lymphocytes are called the lymphoid organs. Origin, proliferation and maturation of lymphocytes are called lymphoid organs. Now, basis of actually the functional aspects or on the basis of functions, we can classify the lymphoid organs into two major groups. One, primary or central lymphoid organs. Two, secondary or peripheral lymphoid organs. What do you mean by primary lymphoid organs? Now, these are the organs where we have the maturation, or we can say the production and maturation of the lymphocytes occur. So, this is the place which provided appropriate environment for the maturation of the lymphocytes. So, those organs which provide appropriate environment for the maturation of lymphocytes are called the primary lymphoid organs. Where we have the production and selection of what we have the lymphocytes. We can say the early selection of lymphocytes. What are the lymphocytes being selected for the functional aspects? Now, under which we have three major organs, two in human beings and one in the case of birds. Now, thymus, bone marrow, these two are the primary or central lymphoid organs in humans. And whereas in the case of birds, we have bursa of Fabricius, an organ present in the case of birds concerned with what we have, that is the primary organs, the primary what is called lymphoid organs. So bursa of Fabricius in birds and thymus, bone marrow in humans together constitute the primary what is called the lymphoid organs. Okay, place of maturation, selection of lymphocytes, etc. Now, what do you mean by secondary and peripheral lymphoid organs? After getting immunocompetent, the ability to compete with what we call the antigens, that process called actually immunocompetency. So, after getting immunocompetent in the primary lymphoid organs, the cells are migrating to the peripheral lymphoid organs where they are anchored. They are concerned with the trapping of antigens and make the antigens available for the mature lymphocytes so that they can attack these antigens. So there is a one what's called secondary lymphoid organs. After getting immunocompetent in the primary lymphoid organs, they are transported to the peripheral or secondary lymphoid organs through the bloodstream where they are anchored, or we can say where they are seeded, another word we can say. And there they are used for trapping the antigens because antigens are localized in the secondary lymphoid organs, so that they are effectively exposed to the mature lymphocytes. So they trap the antigens and make them available for mature lymphocytes so that the mature lymphocytes can fight against these antigens. Now what are the examples? Number one, spleen. Number two, lymph nodes. And these are specific organs. Number three, tonsils. Number four, adenoid. Number five, appendix. They are all abdominal tonsils, articulated. Number six, base patches in the gastrointestinal tract. And also finally, mold. Mucosa associated lymphoid tissue. I'll explain later the expansion. Mucosa associated lymphoid tissue. So these are all the different types of what we call the secondary lymphoid organs, and they are all peripheral in nature, not centralized, hence the name peripheral lymphoid organs. We'll see one by one. First the primary or central lymphoid organs, the examples, the structure and the functions. Then we'll go to the next one, the secondary lymphoid organs. Now, what are primary lymphoid organs? Those lymphoid organs which are involved in production, maturation, and early selection of lymphocytes. These organs are called primary lymphoid organs or central lymphoid organs. So, once the lymphocytes are formed, these lymphocytes become dedicated. So, they are dedicated to actually particular antigenic specificity. So, a particular lymphocyte is attacking only a particular antigen. So, once the lymphocytes are formed, they become dedicated to particular antigenic specificity. Now, in the primary lymphoid organs, the lymphocytes undergo maturation and become immunocompetent. The meaning for immunocompetent, so ready to fight or ready to encounter with the antigen. The ability to encounter the antigen is called immunocompetent, fighting against. So, only when the cells undergo maturation in primary lymphoid organs, then only they become immunocompetent. So, the immunocompetent, actually the cells are educated so that they are capable of just mounting, that is immunity, by fighting against antigens. Mounting immunity by fighting against antigens, and that's the meaning for that one. So they become immunocompetent, the ability to fight against antigens, that is called immunocompetency. So they become immunocompetent in the primary lymphoid organs. Now here there are two different types of cells which are produced by the primary lymphoid organs. One B cells, another one T cells. The B cells referring to the B lymphocytes. As it is produced by the bone marrow, B stands for bone marrow. As it is produced by the bursa of Fabricius in the case of birds, it is called bursal cells. So B stands for bone marrow or bursa. And similarly, the T lymphocytes, they undergo maturation in the thymus gland. So hence they are called thymocytes. T refers to the thymocytes. What is the main difference between these two? If you are taking the B lymphocytes, they undergo production and maturation in the bone marrow. Production and maturation in the bone marrow. Regarding the T cells, the production of T cells during the embryonic stage in the bone marrow, and they are transferred to the peripheral lymphoid organs, like what is called the thymus. Sorry, I made a mistake. To the thymus, not peripheral, to the thymus, where they get mature. Hence, production occurs in the bone marrow during the embryonic stage, but the maturation of the T cells occurs in the thymus, hence called T cells or thymocytes. That's about the differences between the B cells and T cells. We have so many, let's discuss the cell types. Now, under the primary lymphoid organs, we have three examples. Two from human beings. One is thymus, another one bone marrow. The next one, what we got, the bursa of Fabricius in the case of birds. Now we'll take the structural and functional aspects of thymus with illustration, then go for other structures. Thymus is an example for primary lymphoid organ in the immune system. So it is a bilobed structure, a flat bilobed structure, located just behind the sternum, the chest bone, in the upper part of the heart. This is the heart, this is the upper part of the heart. Just behind the sternum, we have the flat bilobed structure. That's the location. If you're taking actually the organ, this is the first organ to be considered as the lymphoid organ in all the species. The first organ to be lymphoid in all the animal species. Now, if you're taking that organ in structural aspect, it is being divided into two compartments, outer compartment and inner compartment. Now, here I given what we have, the cross section. The outer compartment is called as a cortex. The inner compartment is called as a medulla. Normally, it is a bilobed structure. Each lobe is further divided into many lobules by means of septa or trabeculae. Now, the lobules are separated from each other by means of septae, by means of trabeculae or septa. So, here represented the outer capsule which enters into the gland as the septa, So we have many divisions, namely outer cortex and medulla. Normally it is a lymphoepithelial structure consisting of both lymphocytes as well as what we have, that is the epithelial cells. Now if you are taking the outer compartment of the cortex, it is densely packed with immature lymphocytes. And if you are taking what is called the medulla, it is sparsely populated here and there, sparsely populated with mature lymphocytes along with epithelial cells. Some of the epithelial cells get aggregated, what is called hole-like aggregation, just like a pile of points, one above the other, hole-like aggregation of epithelial cell. Here we have hole-like aggregation of epithelial cells, and such aggregation is called Hazel's corpus cell. Hole-like aggregation means arranged one above the other in different layers as a pile of coins. So, in the medulla we have sparsely populated what we have, the mature lymphocytes, and also we have epithelial cells of different types. We have also some cells looking like the nerve cell, and that is called the dendritic cell. Now this is what we have, the dendritic cells, and these are all the epithelial cells. We have what is called the mature and immature lymphocytes. So the thymocytes actually present here, I mentioned already. What will happen here? The cells undergo maturation here. Now the cells, immature lymphocytes, get educated so that they become immunocompetent, mounting immune resistance, able to fight against antigen. That's called immunocompetent. So they're educated in the thymus gland so that they become immunocompetent, able to fight with the antigen. Now, the thymus is also considered as an endocrine gland because it secretes some hormone-like substances. What are the hormone-like substances? The hormone-like substances are thymosin. We have different types of actually hormone-like substances. In the book given only just one, thymosin. This is a hormone-like substance which is responsible for stimulating the immature lymphocytes to undergo maturation. Normally, the gland is growing vigorously, the newborn babies, up to the 12th year. After 12th year, this gland undergoes retrogressive involution. The meaning for the one, it undergoes atrophy, reduction in size, diminution in size. Retrogressive involution, shrinking in size. So it grows to maximum up to 12th year during the lifetime. So the early teens, that is after 12, the early teens, they undergo actually atrophy, and it is simply replaced by, that is what is called the fatty tissues, the adipose tissues. So it is functional, very effective in neonatal period, neonatal and pre-adolescent period. Pre-adolescent period. So it is functional only in the newborn babies. Neonatal means newborn babies, and also in the pre-adolescent period, that is before 12th year, because it is vigorous before 12th year. That is called pre-adolescent period, and becomes atrophy during what is called the adolescent period after the 12th year. So atrophy of the gland occurs when we have the onset of the adolescent period, and also it is decreasing in size, diminishing in size in the adulthood. That's why the immunocompetent power is decreasing in the case of old people, because the thymus gland decreases in size and its function also being decreased. So that's about the thymus gland. So it contains mainly thymocytes, T lymphocytes. Unlike other glands, this gland has its own what is called thyme antigen. For stimulating the lymphocytes production, we need antigen that is coming from outside. But in the case of T cells, thymocytes, if you are taking the T cells, the thymocytes, on the surface we have an antigen, what is called thyme antigen. So it is being stimulated to produce, it is being stimulated to undergo multiplication. by the antigen present on the surface itself. So it is independent of external antigen stimulation. Without external antigen stimulation, now the antigen present on the surface of the lymphocytes stimulate the lymphocytes to undergo proliferation and maturation. So after the lymphocytes produce T cells, only 1% leaves circulation. Only 1% leaves circulation. The remaining 99% of the cells are being destroyed within the thymus itself. That phenomenon is called programmed cell death. That phenomenon is called programmed cell death. Programmed cell death. So it's also aptly called as apoptosis. Technically called apoptosis. So, only 1% of the lymphocytes leave, the remaining 99% of the cells are being destroyed in the thymus itself. That phenomenon is called programmed cell death or PCD. That's technically called apoptosis. So, that's about the thymus, its functional role. Production of lymphocytes, T lymphocytes, and maturation of T lymphocytes. Let's have more information about another primary lymphoid organs, the bone marrow of humans. You know the bone marrow is found in the spongy portion of what is called the bone. In the marrow cavity, we have bone marrow. At the terminal end, you know that one, epiphysis. We have red bone marrow, and then in the cavity, we have yellow bone marrow. It is only the red bone marrow concerned with the production of the cells, not the yellow bone marrow. It is nutritive, rich in fat. Now, the marrow present in the epiphysis of the bone, the terminal end of the bone, contains hematopoietic stem cells. A stem cell is the one which is always having the ability of cell division even in the adult stage. Only the embryonic cells, normally you know that one, they have the ability of undergoing cell division continuously. And we have the stem cells even in the case of umbilical cord, we'll talk about later. It contains stem cells. Nowadays it is possible to produce a new baby from the umbilical cord because it contains stem cells. So stem cells have the ability of undergoing cell division. They have the potential to multiply, to produce different types of cells. Now the stem cells multiply because they have the potential to multiply. Through cell division they can produce a number of cells. Some of the cells remain as stem cells. Some of the cells remain as stem cells. Other cells differentiate and mature into blood cells. Other cells differentiate and mature into blood cells. Different kinds of blood cells. We'll talk about later. Now the process of production of blood cells is called hematopoiesis. What is hematopoiesis? The production of blood cells. The process of production of blood cells is called actually hematopoiesis. That's about the bone marrow. What are the different types of cells we can see under the cell types of the immune system later. Now another one, what we call the primary, that is lymphoid organ, but not in humans, but in the case of birds, bursa of Fabricius. Now it is normally present in the case of birds, present on the dorsal side of the cloaca. Now this is a clavicle aperture. Now this is what is called the cloaca. The region into which the reproductive tract, the urinary tract and the digestive tract open, that is called cloaca, which opens outside through the clavicular aperture. Now this bursa is present on the dorsal side of the cloaca. So in structure, development, embryonic development and function, it is more or less similar to the thymus gland. But it is mostly concerned with the production of what is called the B cells. Somehow it is also concerned with the production of T cells, but mainly concerned with the B cells. So it affords immunity to the body by means of humoral immunity and cell-mediated immunity. Through antibody-mediated immunity and then cell-mediated immunity, it affords protection to the body. That's about the bursa of Fabricius, found only in the case of birds, not in the case of human beings. So altogether we have three primary lymphoid organs, two in man. One is bone marrow, another one thymus, and the third one is in the case of birds, one is called bursa of Fabricius. Okay, that's about the primary lymphoid organs. We have to go for the secondary lymphoid organs. Now, after immunocompetent in the primary lymphoid organs, the lymphocytes are migrated through the bloodstream or the plasma or the lymphatic system to reach the secondary or peripheral lymphoid organs where they are being seeded or anchored. During antigenic stimulus, they exhibit what's called immune response. Now, in the peripheral lymphoid organs, the antigens are localized. So that what will happen, and they are effectively exposed to the lymphocyte. So the lymphocytes can just normally attack the antigens, encounter the antigens very easily because the antigens are localized. This is called antigenic stimulus, which stimulate the immune response. Now, normally they trap the antigens and expose them to the lymphocytes. That is called antigenic stimulus. So when there is an antigenic stimulus, they exhibit what's called an immune response and provide or afford immunity to the body. So we have a variety of secondary lymphoid organs. Number one, the lymph node. Number two, the spleen. These two are the organs. Appendix, tonsils, adenoids, place patches in the gastrointestinal tract. This is what's called actually a system of diffused or aggregates in the gastrointestinal tract concerned with what we have production of lymphocytes. Having different types of lymphocytes it has. And also the marked mucosa associated lymphoid tissue. So out of all the lymphoid organs and the tissues, now major percentage is constituted by the mark. So it provides more than 50% of the lymphoid tissue, of all the lymphoid tissues together. The major one which constitutes the major part, more than 50% is by the marked mucosa associated lymphoid tissue. So we have to go for one by one the structural aspects of lymph node, what is spleen, a few words, then the tonsils, adenoids, place patches, etc. Students, let us discuss about the various types of secondary lymphoid organs in the next class. Thank you.